Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Srivastav. Today we are going to study the topic heterosis. What is heterosis? Heterosis may be defined as the superiority of an F1 which is hybrid over both of its parents in terms of yield or some other characteristics. Heterosis or hybrid vigor describes the superior performance of heterozygous hybrid individuals compared with their homozygous parental inbred lines. In case of hybrid development, we are using two parents. So in such cases, we are selecting the parents because of which we are getting some superior characteristics in its offspring, which is F1 hybrid. Heterosis or hybrid vigor or outbreeding enhancement is the improved or increased function of any biological quality in a hybrid offspring. An offspring is heterotic if its traits are enhanced as a result of mixing the genetic contributions of its parents. So the F1 hybrid's characteristics will depend on its parents. In other way, heterosis also refers to the phenomenon that progeny of diverse varieties of a species or crosses between a species exhibit greater biomass, speed of development, and fertility than both parents. Heterosis term was given by Searle in 1914, which refers to the superiority of F1 hybrid over its parents with respect to yield, growth rate, and vigor, etc. Expression of heterosis is confined to the first generation. Because of this, farmers have to buy fresh seeds every season to raise a commercial crop. The heterosis declines and disappears in F2 and subsequent generations of a cross as a consequence of segregation and recombinations. Because of this, the heterosis expression is mostly limited to the F1 generation. So when the hybrid seed is produced, farmers has to purchase fresh seed to get the maximum benefits of hybrid and getting the good yields. If we are using the stored seed from F1 generation, in next generation there will be drastic reduction of yield and quality. Expression of heterosis is controlled by nuclear genes. Heterosis may be positive or negative, both sides. Both positive and negative heterosis used in crop improvement depend on the breeding objectives. For example, positive heterosis is desired for yield, but we look for negative heterosis also for some traits like days to maturity and plant height. Even though heterosis is used for the best results from hybrid parent. Heterosis, also called hybrid vigor, which results in the increase in such characteristics as size, growth rate, fertility, and yield of a hybrid organisms over those of its parents. Heterosis or hybrid vigor depends on the parental lines used. Hybrid performance is better compared to its parents, so the selection of parents is playing very important role in transmission of heterosis and resulting hybrid vigor. Plant and animal breeders exploit heterosis by mating two different pure inbred lines that have certain desirable traits. Hybrid vigor is used as synonym of heterosis. It is generally agreed that hybrid vigor describes only superiority of the hybrid over the parents, while heterosis describes the other situations as well as like crossing over may result in weak hybrids. If we will go through the clarification between heterosis and hybrid vigor. About hybrid vigor, according to Searle, the developed superiority of hybrids is the hybrid vigor, while heterosis is the mechanism by which this superiority is developed. As we are studying about hybrid seed, hybrid vigor and heterosis, so first we should understand what is a hybrid seed. Hybrid seed is the seed F1 generation produced from a cross between genetically dissimilar parents And the hybrid vigor or heterosis is the absolute phenomenon where expression of the characteristics such as growth related traits, flowering, and yield fall outside the range of its parents with superiority. In hybrid development, we are using two parents, female parent and male parent, and after crossing pollination, the F1 generation resulting is known as hybrid. And the process mechanism by which the hybrid vigor is developed is called heterosis, and superiority of the F1 generation hybrid seed over its parents is termed as hybrid vigor. For the first time in the beginning, the use of heterosis in agricultural practice was carried out on some important crops like first one maize, then on beetroot, Vita vulgaris, then sorghum crop, onion, eggplant, tomato, pepper, rice, cotton, sunflower and rapeseeds. The beginning of hybrid breeding is considered to be in 1920 as per Crow 1998. Use of heterosis in plant breeding. Heterosis is exploited through development of hybrid, and heterosis is commercially exploited in hybrid seed production of cross-pollinated crops of jowar, sorghum, bajra, pearl millet, onion and cucurbits. It has been also used in self-pollinated crops like rice, wheat, tomato and brinjal etc. Now features of heterosis in plant breeding. Heterosis is used for the development of better hybrid lines resulting from its parents. So it is for the development of new lines as per the requirement to get the positive characteristics from the F1 hybrid. The heterosis is having two types: positive heterosis, which is superiority, and negative heterosis, which is inferiority. But these all are used for the benefit of agriculture cultivations. If we discuss regarding positive heterosis, it is the like greater adaptation in wider range, adaptability as per the environment where the crop suits for the cultivation. So we have to generate potential to grow on large commercial scales. Then yield increase in size, enhancing growth, developing disease resistance, insect resistance, improving quality, improving seed vigor and improving reproductive abilities. While in case of negative heterosis, we can go through maturity where we have to develop the short duration hybrids, medium duration hybrids as per the requirements. So in such cases, we are reducing the crop period. After that, plant height. Now the demand is of dwarf hybrids, so we have to reduce the plant height to meet the farmer's demand as per the situation. And another example is reduction in toxic substances. So these are the various types of heterosis. Now we will go through hypothesis of heterosis or theory of heterosis. It is of three types. First one is dominance hypothesis of heterosis, which was proposed by Davenport in 1908 and later supported by Bruce 1910, Kibble and Pellow in 1910. This is the most widely adopted theory of heterosis. And second theory is over-dominance hypothesis of heterosis, which was proposed by East in 1908 and Searle in 1908. Third is epistatic theory. Now we will discuss about each theory in details. First one is dominance hypothesis of heterosis, in which Davenport stated that heterosis is due to the accumulation of dominant genes in a hybrid derived from its two parents. Dominant hypothesis of heterosis holds that increased vigor and size in a hybrid is due to combination of favorable growth genes by crossing two inbred races or inbred parents. Dominant alleles have favorable effect, while recessive alleles have unfavorable effect. In heterozygotes, the harmful effect of recessive gene is masked by its dominant allele. In other words, the hybrid vigor is a result of action and interaction of dominant genes or fitness factors or cumulative polygenic effect of dominant genes. Like example, if we are using two parents for crossing, P1 parent is having the dominant genes of A, recessive genes of B, dominant genes of C, and recessive genes of D, while parents B second is having the recessive gene of A and dominant genes of B, recessive gene of C, and dominant gene of D. So after crossing, the dominant genes will be in effect having the favorable effect, like AA crossing after BB, CC, and DD, where there are 4 dominant genes. So in dominance hypothesis, these favorable dominant genes will having the suppression growth on recessive alleles. Second theory is over dominance hypothesis of heterosis, which was proposed by East and Searle in 1908, which stated that heterozygotes are more vigorous and productive than either homozygous. Overdominance theory considered that there is a physiological stimulus to development that increases with the diversity of the uniting gametes. The overdominance hypothesis is variously known as single gene heterosis, cumulative action of divergent alleles, single gene heterosis, or stimulation of divergent alleles. Fisher in 1930 called it as superdominance. Heterozygotes perform in a given function over a range of environments more efficiently than either homozygotes. Heterozygosity at some loci are superior to both relevant homozygotes, and according to the overdominance hypothesis theory, the result is of superiority of heterozygote over its both homozygous parents. Thus, heterosis is directly proportional to the heterozygosis. Third theory is epistasis, which was proposed by Gowen in 1952. Epistasis stated that influence of one locus on the expression of another may be involved in heterosis. Epistasis refers to interaction between alleles of two or more different loci. Epistasis is also known as non-allelic interaction. It particularly involves dominance effects that contribute to heterosis, and it has been observed in maize and cotton crops. A majority of heterotic crosses show significant epistasis, but all heterotic crosses do not show epistasis, and all crosses that show epistasis are also not heterotic. In general, it can be said that heterosis up to a large extent is due to dominance gene action, but epistasis and overdominance are also involved. Yet the magnitude of epistatic variance is difficult to estimate, and it may play a very important role in heterosis. Epistasis can be estimated by biometrical models. Now, methods of estimation of heterosis. It can be done in 3 ways. First one is mid-parent heterosis, second is better parent heterosis, and third is extended parent heterosis. First one is mid-parent heterosis. When the heterosis is estimated over its mid-parent, like mid-value or average of the two parents, is known as mid-parent heterosis. This is also known as average heterosis or relative heterosis. Like example, if we are using the two parents P1, P2, P1 is having height of 6 feet and P2 is having the height of 10 feet, and after crossing, the hybrid which is produced is having the height of 9 feet. So here the mid-parent average will be taken by making average of P1 and P2, and it will be compared with F1. The calculation formula of mid-parent heterosis is F1 minus mid-parent multiplied by 100 divided by mid-parent value, where F1 is equal to mean of F1 and MP is equal to mean of two parents, mid value of two parents. Second method of estimation is better parent heterosis. When the heterosis is estimated over better parent is known as mid-parent heterosis. This is also known as heterobeltiosis. The term heterobeltiosis was used by Vischer et al. In 1968, which means for the improvement of heterozygote over the better parent of cross. Like example, if you are using two parents P1 and P2, P1 is having height of 6 feet and P2 is having the height of 10 feet, and after the cross the hybrid is having the height of 9 feet. So here the formula for calculation of better parent heterosis is F1 minus BP multiplied by 100 divided by BP, where F1 is equal to mean of F1 means mean height of F1 hybrid and BP is better parent. So here we have to compare the heterosis F1 hybrid with better parent. Third method of estimation is standard heterosis. A standard heterosis refers to the superiority of F1 hybrid over the standard commercial check variety. It is also called as economic heterosis or useful heterosis. In this method, the hybrid is compared with a standard commercial check variety which is prevalent in the market, and the calculation formula is F1 minus check variety into 100 divided by check variety, where F1 is equal to mean of F1 and check variety which is commercial variety prevalent in market and most favorable and adopted by the farmers. So our hybrid is to be compared with commercial check variety for heterosis level. Balanced heterosis versus pseudo heterosis. What is balanced heterosis? A well-balanced gene combinations which are more adaptive to environmental conditions and useful from the agricultural point of view result in balanced heterosis. It has wide applications in hybrid seed production. There is balanced gene combinations and favorable effects are reported, where we can use it for improving the desirable traits in lines while developing hybrids, and it will result in good heterosis for improving the yields, maturity and whatever the desirable characteristics are needed. Now pseudo heterosis, these are also termed as luxuriance, where progeny possess superiority over parents in vegetative growth but not in yield and adaptation, and these are usually sterile or poorly fertile. This concept cannot be utilized in hybrid seed production. So here in case of pseudo heterosis, this is related to only vegetative and morphological growth but not on yield attributing characteristics. So this is not desirable for hybrid breeding. Now factors affecting magnitude of heterosis. There are 4 genetic factors which are affecting the magnitude of heterosis. Like first one, mode of pollination. There are 3 modes of pollination methods in crops: self-pollination, cross-pollination and off and cross-pollination. So these pollination methods are affecting magnitude of heterosis. Second is genetic diversity of parents. While hybrid seed production, how the parents' diversification natures are utilized in hybrid seed development is impacting the magnitude of heterosis. Third is genetic base of parents, means where in case of hybrid seed development, the parents are playing very important roles to insert the desirable traits. So their genetic base which will play very important role. Fourth is adaptability of parents in different agroclimatic conditions and diverse nature. So all these factors affect magnitude of heterosis. So this presentation was all about heterosis. Hope this will be useful to all of you. Thank you very much. I have given here my YouTube channel details, Journey with Vijay Kumar Srivastav. Having request, please visit the channel, subscribe it, and provide your kind and valuable feedback. Thank you.